Let's rock and roll. Okay, so the idea here is that we are going to pick up where we left off on Thursday of last week versus Thursday of any other week. Um, and then we'll get as far as we can today. Okay, so last week we started working through the first few classes of enzymes, right? We focused in on oxidoreductases and we made it all the way through our review of oxidoreductases. The important thing to remember is number one, that so very many enzymes that we see this semester are oxidoreductases. Anytime you're dealing with electron transfer, we are talking about oxidoreductases. And the two really kind of big recurring themes that I hope you picked up and remember is number one is that the cofactors we deal with tend to repeat. The two big ones are ADH and FADH2. At least when we're talking about organic molecules and electron transfer. Whenever we're dealing with organic molecules and electron transfer, you should be thinking about one of those two, with a couple exceptions, right? When we get to the electron transport chain, we'll talk about coenzyme Q, and you even saw it in the activity, or I guess not the activity, but the lecture on Thursday. But for the most part, when we're dealing with those organic molecules, you shouldn't be thinking about individual electrons. Always think about hydride transfer, right? Whatever the electron donor is, it gives electrons away in the form of hydride. Remember, hydride is simply a hydrogen anion. You never think about single electrons hopping from place to place, except in the electron transport chain. Any other time, you're thinking about producing or consuming a hydride anion. Okay, in the enzyme that y'all saw in biochem lab, lactate dehydrogenase, right? The enzyme used lactate as an electron source, and it generated hydride. It transferred that hydride to NAD+. Okay, so you should be familiar and comfortable with the two cofactors NADH and FADH2 and how hydride moves through those cofactors. If you're comfortable with that, then every other oxidoreductase will just kind of make sense. Once you generate hydride, you know what to do with it. If you need hydride, you know where it comes from on those cofactors. Be comfortable with that. Okay. The other kind of big theme is that if you look at a table of reduction potentials, you should always be able to very quickly, without doing any math, predict whether a reaction will be spontaneous in one direction or another. It's all about the reduction potentials. If the reduction reaction has a more positive potential than the corresponding oxidation reaction, it will be spontaneous every time. Okay? It's just a matter of thinking about their position in that table. All right, so today we're going to kind of move on and continue our discussion of transferases. Last week we did get through a lot of the methyl transfer strategies we talked about. We saw that THF can come in a couple forms, right? It can come in a form where you have a methyl group sitting there, a formal group attached to the nitrogens, or a methylene. So it's an important cofactor in that it can transfer carbon in multiple different forms, okay, multiple different oxidation states. And we also saw SAM, which is S-adenosylmethionine, not you. You weren't here. You missed it. S-adenosylmethionine, which is an adenosine covalently attached to the sulfur of methionine. And when that happens, that last methyl group that's attached to the thioether side chain of methionine is very reactive because that sulfur doesn't want a positive charge. So those are the two cofactors that very commonly transfer methyl groups in our reactions. Okay? And then we started introducing the transamination reactions. Okay? And this is what we're going to use to represent how another really important cofactor, PLP, works. PLP is pyridoxal phosphate. And the idea is this cofactor is an incredible electron sink and facilitates the breaking of bonds that we otherwise would normally think aren't necessarily labile immunity reactive. Okay, and we started using this transamination reaction as an example. So with all of these reactions, kind of what we're thinking about, at least these transamination reactions, what we're thinking about is starting off with one amino acid and one alpha keto acid. And remember last week I mentioned that the alpha keto acid is, so if this is the highest priority carbonyl, which it is, our carboxylic acid, the alpha position simply has this ketone. So alpha keto. We're going to see lots of alpha keto compounds. This is the first example this semester. So the idea is the two reactants are an amino acid, it can be any amino acid, and any alpha keto acid. Okay, different enzymes evolve to have different amino acids or different alpha keto acids as substrates. But the idea is basically we swap positions. What you'll note here is on the product, our alpha keto acid, it is the original amino acid, but instead of having this amine here, we now have our alpha keto. And what was the alpha keto acid became the amino acid. So these transaminases always catalyze the swapping of the amino group with the oxygen group, always on the alpha position. Pretty straightforward. And the way that it works has to be in a way that does not evolve ammonia. Okay, ammonia is a molecule that in really small amounts isn't a big deal because we have buffers in our cells. It's not a big deal. We can take care of the production of this base. But because amino acids are in such high concentration in the cell and we need to recycle them all the time, if there was a strategy to produce an amino acid that involved the evolution of ammonia, even temporarily, it would be pretty dangerous because we would have a massive surge of ammonia at any given time, and that could aggressively influence the pH and therefore everything else in the cell. So the idea here in this kind of mechanism that I'm showing here is that we start with one reactant, our amino acid, and our other reactant, the alpha keto acid. This reaction overall is kind of two phases. In the first phase, the amino acid reactant loses its ammonia and is replaced with water to produce the alpha keto acid. In the second phase, the ammonia reacts with our second reactant, in this case the alpha keto acid, and produces the amino acid. So this reaction, no matter how you draw it up, has to happen in two phases. And the transition between these phases evolves ammonia. The evolution of ammonia by itself, freely floating in the cell, can't happen because it's dangerous. Okay, and we're going to see other examples of this later in the semester. So the idea is pretty simple, is that we have to develop a mechanism that avoids this complication. And that's where our cofactor PLP comes in. Okay, so we're going to look at this cofactor in pretty decent detail here in the next few minutes, but the idea is reasonably straightforward. The pyridoxal phosphate itself comes in the form of having a ketone here at this terminal position. Or it's not a ketone, it's an aldehyde at this terminal position. Okay, this is the way we eat vitamin B6. This is PLP. Okay, when it comes into our cell, the first thing that happens is it reacts with the active site of an enzyme, and it gets anchored into the enzyme through the formation of a shift base at this position, which we'll see a couple times here in the next few minutes. The beauty of that is that it allows us to create this intermediate, this pyridoxamine phosphate, where at this position we have the amino group covalently modified, covalently bound, and therefore it's not freely evolved to float around the cell. So it's this form of our cofactor that allows us to prevent the evolution of this nitrogen, of this ammonia group. Okay, let's look at how it happens. All right, so I mentioned that all of these cofactors, PLP always, is originally anchored in the active site of the enzyme through the formation of a shift base. And if you're not familiar with what a shift base is, it's simply an imine linkage, right? So it's the nitrogen here double bonded to a carbon, and that gives you this positive charge, okay? That positive charge is a very beneficial thing, okay, because it makes this nitrogen a good electron sink. And when you took it organic, if you didn't hear the term electron sink, all that means is it's a really good place to dump electrons, okay? So this nitrogen in our shift base is a good electron sink. In addition, and what we're going to see much more importantly, is this nitrogen at the base of our cofactor is an excellent shift base, or excellent electron sink. And that's going to become really important in the reactivity of this cofactor. Okay, so Once the cofactor enters our cell, it finds an enzyme that uses it, and the first step is always the formation of this shift-base linkage between the cofactor and a lysine side chain in the active site. This ensures the cofactor is anchored at the right place. It can't diffuse out. Okay? And then in the first step of every single PLP dependent reaction, the first step is your substrate amino acid comes in and it displaces the shift base between the cofactor and the lysine side chain, right? So we have lysine in the active site now with a free amine, and the shift base is now formed between the amino acid and the cofactor, right? So this is the new shift base. Our substrate is now covalently bound into the active site through the formation of the shift base with the cofactor. This is a critical step, okay? Because once this happens, what you can't really see in this view unless you think through it really clearly, but what you end up seeing now is that one of these bonds is incredibly reactive because of this pi system, right? So if we go back here one step and look, in our cofactor, we have sp2, 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 and then every atom in our benzene ring, in our ring system, is sp2 hybridized. So there's this conjugated pi system that carries through all the way up to our sp3 alpha carbon. One of these bonds on our alpha carbon is in the plane of the pi system, and it all depends on how the amino acid is rotated in the active site, right? So this nitrogen is our electron sink. Screw you. So this nitrogen right here is our electron sink. And the idea is one of these bonds is parallel to our pi system, right? So we can see in this case, it's this bond right here between the alpha carbon and whatever this R group is. That makes this bond really, really reactive, okay? The bonds that are not in this pi system, not in the plane of the pi system, are not reactive. So the beauty of this idea is that if you can look into the active site, right, you have the crystal structure of these things, and you can look into the active site, you can very quickly tell which bond is going to be reactive, which bond is going to be labile, because it'll be the only one that's close to parallel to the pi system, right? So within the pi system, okay? And that's the bond that's reactive. Okay, so if we look at two different PLP-dependent reactants. So in this one right here, this is aspartate transaminase. This is one of the enzymes we talked about that we're going to look at. That basically the idea is you swap the amino group with the aspartic acid and then something else. We see here that it's the CH bond on the alpha carbon that's perpendicular to the plane of the system, to the plane of the cofactor. In another one, the serine hydroxymethyltransferase, this is important in serine metabolism, we see that it's the bond between the alpha carbon and the beta carbon on the side chain. So in this enzyme, it's actually going to be this bond that's reactive that ultimately we might break. Okay, so you can tell which bond is reactive based on its position. And it's all about which one kind of sticks out of the plane of our cofactor. And we won't go into the chemistry of that one in much more detail, but just an example of what you can look for. All right, so let's see how all this works. So over the next few slides, it's set up so that you can follow along on your activity. So the last 2 pages of activity, just follow right along. I'm going to walk through the mechanism. You can follow along. Okay? And I'll do my best to explain why things are happening and what's happening and why and how. And if you have questions as you do it, ask me. This is the time to ask, okay? All right, so in step 1, we're explicitly said the amino acid enters the active site and swaps place the lysine, okay? And usually when I ask you to draw these out, especially if it happens to be on an exam or something that's time sensitive, I usually am perfectly fine with you just saying that they swap, okay? But for the time being right now, we can do this chemistry pretty easily. In our reactants, we see that we have the shift base with our lysine side chain. In our products, we see the lysine has been released, and we have the shift base with the amino acid. Okay, so we know that we need to make a bond between this nitrogen and this carbon. So this carbon becomes our electrophile. It's electron deficient because of its bond with the nitrogen on the lysine. Simply make a bond and imagine that there is a hydrogen donor, a proton donor. So you just kind of pick up that proton. Actually, let's just skip that part. Okay, we'll put the electrons up onto this nitrogen. So the idea here is we form a pretty simple intermediate. We have our lysine still bonded to one hydrogen. It has a lone pair. Bonded to the rest of the cofactor, I won't draw it. And then this one is bonded to two hydrogens and then the rest of our cofactor, or the rest of our amino acids. So here's the alpha carbon attached to R, attached to H, attached to CO2 minus. Okay, so I simply drew an intermediate. Okay, this nitrogen was the nitrogen that was part of the shift base. It now is single bonded to our central carbon. This central carbon is attached to the rest of the cofactor and the nitrogen from the amino acid. This should have a positive charge on it. So this nitrogen now has a positive charge. This is the nitrogen on our amino acid. From here, we need to break this bond, and we need to end up with a double bond here. So imagine it however you want. I'm going to just kind of say that maybe we do a simple proton like cycling, a kind of swapping. You can have it as a general acid, general base, however you want to do it, however you want to think through it. But for my purposes, I'm just going to say that this lone pair reaches out and picks up a proton from somewhere. And just for simplicity's sake, I'm going to say it picks it up from the nitrogen on the amino acid. That's probably not what actually happens. There's probably a general acid around there. But for our purposes, this works great. Make a bond, break a bond. We made a bond, so now we need to break this bond. We're going to make a double bond between the nitrogen of our amino acid and our central carbon on our cofactor. Right? We know we have to make this because we end up with a double bond here. We know we have to break this bond, so let's then move these electrons up to here. Pretty straightforward, right? We need an intermediate here because we're creating two bonds and we're breaking two bonds. So one step, we make and break one bond. In the second step, we make and break the other bond. And we end up with our shift base now between the cofactor and our substrate amino acid. These two steps, the formation of our intermediate where everything is bonded together and the release of the lysine and the formation of the shift base, these two steps happen in every single PLP-dependent reaction. The amino acid is the first substrate. It comes in, it replaces the lysine every time. Okay? Come on. Oh, for fuck's sake. Okay, so I like how I record myself saying for fuck's sake. Just realized that. Okay, so we've released our lysine. We now have the reactive form of our amino acid. Okay, so let's kind of think about what we need to happen here. So we have our cofactor. It's covalently bonded through this shift base to our amino acid. And what we see here is that in the first, in this step, we have a general base that gets protonated. And the proton comes from the alpha carbon. And remember when we looked at the structure of this transaminase in the active site, we saw that it's the CH bond on the alpha carbon that was perpendicular to our planar cofactor. That makes this bond reactive. Okay, so the idea here is that we have our lone pair on our general base. Also remember this nitrogen is an electron sink. And the way this cofactor works almost always is electrons are going to go down and flow to this electron sink so they can be temporarily stored. This bond is reactive because it's linked electronically to this nitrogen. Okay, so the base picks up a proton. We move electrons all the way through our cofactor, all the way down to our electron sink. We have now broken the bond between our alpha carbon and that hydrogen. And the electrons are situated happily down on that nitrogen that was our electron sink. So we've just fiddled with the resonance form of our cofactor. Okay? In this step, we still have that lone pair there on our electron sink. what we notice here is that I'm highlighting a general acid in the active site. Nothing is happening up here, right? So everything up here looks the same. Nothing is happening. So everything is happening now with the cofactor. And what will the big thing we notice is that this carbon starts sp2, ends up sp3, and our general acid ends up a general base. So hopefully it's fairly clear for you to see that this sp2 hybridized carbon picks up the proton from our general acid, giving us our other resonance form of our cofactor. So let's just move electrons up through the ring. These electrons in the double bond, in the pi bond, will reach out and become a sigma bond with that hydrogen. We have now deprotonated our general base, and we have an sp3 carbon here that's attached to the nitrogen on our amino acid. Question so far? Okay. The fourth step, and really the last step in the first phase, is first we hydrolyze the Schiff base by bringing in hydroxide. Okay, that hydroxide, of course, comes from water, right? So within the active site, something deprotonates water. We have hydroxide. So we're explicitly told that hydroxide is a reactant. So let's just draw it. Okay. What we see in the first step here in this in this step is that the hydroxide is going to end up covalently bonded to our alpha carbon. Okay, and we also see that our nitrogen goes from sp2 to sp3. So the electrons are going to end up on this nitrogen. Since it's a Schiff base, it's a good electron sink. So we move the electrons here, push them down onto that nitrogen. We form our intermediate. In the next step, what we notice is we bring in a general base, and this base gets protonated, and we see that we break the bond between the alpha carbon and our nitrogen, and this oxygen ends up as a double bond, right? So we end up with a carbonyl. Okay, so the lone pair on our general base picks up the proton, push the electrons through, make our carbonyl. We have some sort of a general acid down here that's going to donate a proton. We'll break the bond between our alpha carbon and our nitrogen. The general acid gets deprotonated. That gives us NH2, and the alpha keto acid is released. Our general acid up here is protonated. Okay, so before we move on, I want to ask, does anybody have questions about these 4 steps? If you're comfortable with these 4 steps, the next 4 steps are exactly reversed. This is the first phase of these reactions. In the first phase, we have an amino acid that becomes an alpha keto acid. And we also evolve ammonia. That ammonia is stored right here on our cofactor. So in the next 4 steps, we simply reverse it. Step 5 is exactly reverse of step 4. We have a new alpha keto acid that comes in. This is a different one. What we see in the first part is this alpha keto acid gets covalently bonded onto our cofactor. And then ultimately here, we're going to evolve hydroxide. I'm not going to walk through all this with y'all. Y'all can work through these 4 steps yourself because they're exactly reverse. Okay, so in the first part of step 5, make the bond. This nitrogen is reactive. It's your nucleophile. The alpha carbon is your electrophile. Make the bond, pick up a proton. In the next step, evolve hydroxide and create your Schiff base. Okay, in step 6, this is exactly reverse from step 3. Remember in step 3, we protonated this carbon in our PLP cofactor. So in this step, we have an sp3 hybridized carbon here. It becomes sp2. We start with the form of our cofactor where we have a positively charged nitrogen down here, and we end up with the neutral sp3 hybridized. So you'll push electrons through to make that happen. Step 7, perfectly reverse from step 2. This is where we are fiddling around with our alpha carbon now. We start with an sp2 hybridized alpha carbon because it's shift-based to that nitrogen. We have a general acid that becomes a general base, and we have one form of our PLP cofactor with a lone pair on our nitrogen, and it ends up as the positive charge. So start with these electrons, push them up through the cofactor all the way up to the alpha carbon, and protonate that alpha carbon using the general acid. And that creates your amino acid. The amino acid is made. It's just anchored onto the cofactor with the shift base. So in the last step, this is kind of that two-phase process where first you use the nitrogen on your lysine side chain to create a bond with that central carbon, and then break the bond with the amino acid. Evolve your amino acid. Okay? So we walk through the first 4 steps explicitly. Remember that the last 4 steps are simply, truly just reverse of that. We're using a different alpha keto acid as a substrate, meaning we produce a different amino acid as the product. That's how all of these transamination reactions work. Yeah. So if we go back to step 5, right? So step 5 here, the reactant of step 5 is this alpha keto acid and our PLP cofactor that has this nitrogen, right? If we go back to step 4, that's really exactly what we produced. In step 4, we produced this alpha keto acid and we produced this ammonium, like this nitrogen that's anchored to our cofactor. So we start, this is where we end in step 4, and that's where we begin in step 5. So if you go back, and if you don't have the activity in front of you, then go back and just kind of like look after class at your activity. What you'll see, compare step 5 and step 4. They are perfectly reversed. The products of step 4 are the reactants of step 5. Compare step 6 and step 3. Compare 7 and 2 and 8 and 1, and you'll see that they're genuinely the reverse of each other. Okay? Any other question?